lung cells from oxidative stress. Therefore, our central research question was very simple, whether CAPE could mitigate or attenuate the cellular damage induced by fructose exposure. For our experiment, we used Syrian hamster pancreatic beta cells. It is a very widely used model of diabetic research. So our experimental design consisted of three main phases. First, we cultured the cells and then exposed them to high fructose concentration to induce oxidative stress. Then CAPE was applied. Second, we measured oxidative damage using T-bars and protein carbonyl assays, which quantify lipid and protein oxidation. In the third phase, we performed gene and protein expression analysis to examine how CAPE influences molecular pathways involved in oxidative stress and inflammation. Together, these approaches allowed us to evaluate both cellular damage and protective mechanism. So now I'll move to the result section. So this slide shows the result of our T-bars assay. Malondialdehyde levels increased significantly in cells exposed to fructose, indicating elevated oxidative damage. However, CAPE decreased the level significantly. So this result demonstrates that CAPE effectively reduces lipid peroxidation and protects cellular membranes from oxidative damage. Next, we did carbonyl assay to measure protein oxidation. Fructose exposure caused a significant increase in protein carbonyl levels, and CAPE could reduce it significantly. So this result indicates that CAPE protects proteins from oxidative damage and maintains cellular integrity. Next, we examined gene expression analysis of two important antioxidant enzymes, GPX1 and catalase. Fructose treatment significantly suppressed GPX1 expression, weakening the cell's antioxidant defense. However, CAPE treatment restored the GPX1 expression toward normal levels. Even more interestingly, CAPE significantly upregulate the catalase expression beyond the control level, suggesting an enhancement of the cell's antioxidant defense system. Therefore, CAPE not only reverses the fructose-induced suppression of fructose-induced suppression, but enhances the endogenous antioxidant defense system. We also investigated hypoxic stress response pathway. High fructose exposure induced a strong increase in interferon alpha and beta expression, creating a pseudo-hypoxic state within the cells. However, when CAPE was introduced, expression was reduced dramatically. So this suggests that CAPE helps maintain cellular oxygen balance and metabolic stability under stress condition.